Bismillahirrahmanirrahim. Assalamu alaikum to all our viewers. This is our educational channel Learning Biology Online, and I am Dr. Asad, and with my specialty within the subject of biology I will be discussing some very important chemical substances that impact the enzymatic activity known as the inhibitors. And we will also discuss the classification and try to understand how different inhibitors they impact the enzymatic activity. End of this lecture, we hope that you will be able to define inhibitors, classify them into different types on the basis of their function, and simultaneously we will be able to understand कि किस तरह से inhibitors जो हैं they are distinct from each other and how they perform their activity. Starting off with the definition, the inhibitors they are chemical substances that bind to the enzyme at the place of the substrate, and they block its activity by temporarily or permanently binding to its structure. तो substrate तो obviously products में convert हो जाता है, but what the inhibitors do, they bind to the active site or maybe any other area of the enzyme structure and they prevent its activity. Coming to the classification, we have a group of inhibitors known as the irreversible inhibitors. तो इनके नाम से ही हम उसके mechanism का idea लगा सकते हैं कि they do adhere to the enzyme globular structure at the active site, but what they do is that they form a very strong bond at the active site, due to which the enzymatic activity is blocked permanently. So in order to understand this mechanism, we will just have a review of the globular structure of the enzyme. So this is how it look like. So एक region के ऊपर we have an active site at which the substrate is going to attach for its conversion into products. But what happens over here is that sometimes the inhibitor, it may attach to this surface and it covers the active site. So this time an enzyme inhibitor complex is formed at this particular area and the enzymatic activity is blocked. There is no formation of any product. तो ये जो irreversible किस्म की inhibitor की attachment हुई है जिसकी वजह से irreversible inhibitor अपना function perform करते हैं. This is a very strong bond between the inhibitor and the enzyme complex, and the bond, this is the covalent bond that exists between the inhibitor and the active site of the enzyme. Then we have a group of enzymes that are labeled as reversible inhibitors. So the reason why they are known as irreversible because they bind very loosely to the enzyme structure, which means that their impact can be reversed, maybe by addition of further enzyme or some other substrate that may come and attach to the enzyme later on, and it can replace the inhibitor effect. So there is a weak formation or association between the enzyme and the inhibitor that can be reversed in this case. Then we come to the competitive inhibitors. इसमें क्या होता है कि जब भी एंजाइम का एक मॉलिक्यूल है वो इनहिबिटर्स के साथ इस स्पेस के ऊपर अटैच हो जाता है बिकॉज इट रिकग्नाइजेज इट एज अ सबस्ट्रेट, लेकिन जब ये बाइंड कर जाता है competitive inhibitor बिकॉज इट इज़ एज competitive एज अ सबस्ट्रेट, तो ये फ़ौरन से आके एंजाइम के ऊपर जब अटैच हो जाता है तो ऑब्वियसली ये एक्टिव साइट को ब्लॉक कर देगा टू कन्वर्ट इनटू प्रोडक्ट्स, जिसकी वजह से जो इन competitive inhibitors हैं दे आर रिकग्नाइज एट द बाइंडिंग साइट, जिसकी वजह से एंजाइम की एक्टिविटी जो है वो कॉम्प्रोमाइज़ हो जाती है. सो इसमें और रिवर्सिबल में यही फ़र्क होता है कि इसमें जो बाइंडिंग साइट है दैट रिकग्नाइजेज the inhibitors, due to which the inhibitor attaches to the surface of the enzyme, and finally the products are not formed and the catalytic site is also not developed or activated. Lastly, we have the non-competitive inhibitors. So they attach to the enzymatic activity एट अ प्लेस अदर देन द एक्टिव साइट। तो जैसे कि ग्लोबुलर स्ट्रक्चर हमने पढ़ा हुआ है एंजाइम का, तो ग्लोबुलर स्ट्रक्चर के किसी भी एरिया के ऊपर ये जो इनहिबिटर्स हैं ये अटैच हो जाते हैं एंड दे halt or inhibit the activity of the enzyme. So this is the end of our lecture where we have discussed the basis of inhibitors and what different types of inhibitors they can impact the enzymatic activity. So do like our video if you have had a good understanding about these chemical substances and their impact, and do share it with your colleagues. In the comment section, do ask questions that you have relevant to the topics that we have covered in this video, and do subscribe to our channel and click the bell button so that you can receive the upcoming video lecture notifications on regular basis. Thank you very much.